Hello, everybody, and welcome to the Frontiers in Science Deep Dive webinar on breakdown and repair of metabolism in the aging brain. I am Laure Sonnier. I am the executive editor of Frontiers in Science. So today's webinar presents the Frontiers in Science lead article led by Professor Henry Markram and colleagues on the most comprehensive computer model of brain metabolisms to date. So I'd like to thank Professor Markram, Dr. Shichkova, Dr. Keller, and Dr. Coggan, who are all here with us today for bringing this important work to Frontiers in Science. We are delighted to give to this article a home in our journal. So before we meet with all these authors and our guest experts today, I'd like just to give you a few words about Frontiers in Science. So Frontiers in Science is our flagship multidisciplinary open access title at Frontiers. The journal focuses on transformational science to accelerate solutions for healthy lives on a healthy planet. So the way we work is that we commission exceptional peer-reviewed articles from world-leading researchers. And then around each of these articles, we built a unique hub of multi-audience content, including viewpoints, articles, editorials, policy articles, and content also for children and the general public. This is all designed to extend the journal's reach and impact across science, policy, and society. So to date, no other publisher does this in the way we do. And we are really delighted to be driving innovation in publishing so that this type of critical, transformational, open science accelerates scientific progress for all. So today's discussion is going to be moderated by my colleague Lee Baker. Lee is the editorial advisor at Frontiers in Science. Hi, Lee. I'm delighted to hand over to him now. Thank you all for being with us. Enjoy the event. And bye for now.
Thanks, Laure, and hello, everyone. This deep dive today is going to be focusing on this lead article, which was published back in March. That's breakdown and repair of metabolism in the aging brain. As you'll be aware, the rise in neurodegenerative diseases is a major healthcare and social care challenge worldwide. And in recent years, the role of brain metabolism in the pathophysiology of these diseases, including dementia, has been highlighted. And so this article presents the most comprehensive model yet of the neurogliovascular system, the NGV system, integrating key cellular and subcellular systems together with molecules, metabolic pathways and processes required to link neuronal electrical activity with brain energy metabolism and the vasculature. The new model has provided open access to help scientists identify and predict and test potential ways in silico to protect brain function during aging. For example, via new drug targets, via dietary interventions or exercise. It's already had a lot of attention. We've had over 72,000 views, as you can see on the top of the screen there, and over 11,500 downloads in just 6 weeks. The work was conducted as part of the Blue Brain project, led by Professor Henry Markram, and he's here today together with the co-authors Polina Shichkova, J. Coggan, and Daniel Keller. This is the article in the hub that Laure referred to. This is the article hub that you can find online. And it's published together with an editorial by Professor Mark Mattson, who's here with us today, a viewpoint by the peer reviewers, Richard Buxton and Luc Pellerin, and a policy outlook by Professor Jolivet. Renaud Jolivet and colleagues at Maastricht. You're going to also find a lay explainer. Here's the rest of the materials. These are designed to make sure that this science is, I beg your pardon, this is designed to make sure this material is viewable by everyone. And finally, yep, just running through. Sorry about that. A little glitch there, and also an article in our journal, Frontiers for Young Minds, to make sure that even future generations can see and understand and be inspired by this new work. So next slide, please. Here is the order for today. So first of all, Professor Henry Markram from the EPFL, the École Polytechnique Fédérale de Lausanne, and the Open Brain Institute will lead off. He'll introduce us to this article and particularly the concept of digital brains. Then Dr. Polina Shichkova from Biognosys in AG in Switzerland will discuss the key findings and the methods behind the model, together with Dr. J.S. Coggan from Neurolinx in the USA. Next, Dr. Daniel Keller will talk about how this model can be used online at the Open Brain Institute using a Jupyter notebook. And then we'll move to a panel discussion and an audience Q&A where we'll be joined by Professor Mark Mattson from Johns Hopkins in the USA and by Professor Pierre Magistretti from the King Abdullah University in Saudi Arabia. So over to our first speaker today. I'm delighted to welcome Professor Henry Markram. Henry is a world-renowned neuroscientist whose pioneering work has helped to reshape the landscape of brain research and particularly computational neuroscience. At EPFL, he founded the Brain Mind Institute and the Blue Brain Project, a Swiss national research infrastructure project, which has established the role of computational simulation neuroscience in concert with theoretical, experimental, and clinical neuroscience research. He's also founded the Human Brain Project, a flagship initiative of the European Commission. Henry is now president of the newly launched Open Brain Institute, a non-profit organization whose mission is to empower researchers with advanced digital brain building technologies, accelerating and democratizing neuroscience research, embracing open science and global collaborations. Henry is also co-founder and chair of Frontiers, the Frontiers Research Foundation, and the Frontiers Planet Prize. So, Henry, thanks for bringing this important piece to Frontiers in Science. We look forward to your introduction. The floor is yours.
Wonderful. Thank you, Lee. Thank you, Laure. And great to be here. I will just give you the context in which we did this study on the NGV with Polina as the lead author and a number of other authors. So the context in which we did this is the Blue Brain Project. And just briefly, the Blue Brain Project is a project that was, we were put on a mission about 20 years ago. The Swiss government gave us about 300 million and 20 years to find the recipe how to build the brain, step by step. It's not so much building a model of the brain, but an actual recipe of how would you go about building the brain, and in principle, any brain. So we practiced on the mice. We also practiced on some parts of the rat brain and also practiced on some parts of the human brain. And the goal of this is not a conceptual model or abstract or theoretical model of the brain. It is a replica. It's a digital twin, which means our goal is to have the algorithms that allow us to recreate the brain in digital form as close a copy as the real brain in terms of the number of neurons, the locations of the neurons, the shapes of the neurons, the behaviors of the neurons, the connectivity, the connectome, the synapses, and also how they all work together. We completed this project in December last year, and we've moved the recipe, which is 18 million lines of code, into the Open Brain Institute so that everybody can build brains. So you can get a virtual lab and you can build neurons or circuits and you can run these algorithms, and you're enabled by AI so that it makes it very easy. Now, 18 million lines is a lot, and to make it easy and accessible for everybody to be able to build brains, we simplified the process into 5 basic steps, and I'll just quickly run you through these steps. The first step is, of course, you get an atlas. There's an atlas of the mouse. This is the mouse brain, which has about 600 brain regions, and you fill it with neurons. But we have AI that reads the literature, goes to databases, it mines the data on every study that has mapped out the number of neurons in every brain region. And you fill it with neurons, but you also tag each of the balls, the dots, with the type of neuron that is known today. You get comes from data in terms of the cell types, morphological types, electrical types, and so on, which comes from a wide range of data, all the way from gene transcription data to staining, immunohistochemical staining, to counting, and so on. So the first step is you've got to get your neurons. The next step is you have to grow the dendrites. Now, when we started, we reconstructed them under a microscope, but what we solved was the mathematics, how to recreate mathematically, how to grow the dendrites. So we can now grow. This was an unsolved mathematical problem for about 50 years, and we solved this, and you can now grow the dendrites of any cell type in the mouse brain and any other brain. You feed it with a few statistical features of an existing neuron that you can get from an image or a drawing, and it can recreate that neuron in a way that's quite indistinguishable even by the most expert anatomist. So once you have your dendrites, you need to get your axons. Lots of axons, very, very long. I mean, you know, your brain is a shocking highway of cables. This was a much more difficult problem because that's really what we call the projectome, where the axons have to project across the entire brain, especially the pyramidal axons or the excitatory neuron axons. But in the subcortical regions, they also have to project, such as in the substantia nigra, they have to project up. So we now know how to grow all the axons of every type of cell in the mouse brain. Now, once you have your dendrites and your axons, you have to connect them. So you basically identify where they are touching. And we have data that shows that this is actually the primary rule for how the connectome arises. It's not the only rule. There are a lot of biological principles that decide, even if an axon touches a dendrite, that it makes a connection or not and forms a synapse. So we have mined and identified all the major rules. There are many still that we can refine, but most of the major rules that decide which subset of all the touch points become synapses. And in that way, we derive the entire connectome. It is an algorithmic derivation of the entire connectome. We've tested this extensively against data in the literature. We've published most of this in about 300 papers in the course of these 20 years in the blue brain. In the step 5, we have to switch it on. So that means every type of neuron has to be modeled mathematically. They are modeled with Hodgkin-Huxley equations. They're biophysical models. And each cell type, there's different electrical types. There's bursting cells, regular spiking cells. And we recreate the electrical behavior of any of the cell types. And for all the cell types in the mouse brain, about 70 million of them. And they form a vast number of synapses. And you need quite large supercomputers, but we've now moved that to the cloud, so you can actually perform these simulations in the cloud, in the virtual labs. Now, to introduce the talk, we not only recreated neurons, we actually worked with Pierre Magistretti, who's one of the panelists in KAUST, to get electron microscopic images of the astrocytes. And then we digitized these astrocytes, and we learned the mathematics of how we can grow lots of astrocytes. It turns out that there's about, in the neocortex, 1-to-1 relationship. So if you have 3,000 neurons, you'll have about 3,000 astrocytes as well. So the blue here shows the astrocytes. On the left, the yellow shows the cortical column with dendrites and axons. And on the next one, the blue one shows the actual the astrocytes. And then we also reconstructed the entire vascular system of the whole mouse brain. And this is a part, which is a small sliver of the neocortex. And then when you merge them together, similarly to when you form connections, you form the touch points between the astrocytes, the vasculature, and the neurons. And you form end feet, and you create a very realistic model with nanometer precision of how the astrocytes are touching the blood vessels in order to be able to support the neurons. So this involves, that's the reconstruction of the vascular system of the whole mouse brain. And here you can see a zoom into where there's a neuron and you have axons and you have the astrocytes touching onto the blood vessels. And now the study that we did was to go even deeper, which is to go into the molecular domain and to try to identify all the factors involved in metabolism, the transporters, metabolites, and all the factors, ion channels, that are involved in creating the metabolic system. There's a lot of data out there on the metabolic system, but it was not all put together. In fact, the most pioneering studies were done by Pierre Magistretti and Jean Olivier and many others. But there was a huge amount of data. And so in this study, we gathered everything that we could find about the metabolic system, and we recreated that. And Polina and Dan and Jay are going to tell you what we found when we created and simulated the brain's metabolic system for the young and for the aging brain. And over to you, Lee.
Wonderful. Thanks, Henry, for that beautiful introduction, setting the scenes for today's deep dive into the model itself. And on to our next speakers, Dr. Polina Shichkova and Dr. Jay Coggan. Polina is currently bioproteomics application data scientist at Biognosys in Switzerland. Formerly, she was a postdoctoral researcher at the Blue Brain Project at the EPFL. Her research centers on computational systems biology with a focus on molecular neuroscience, brain aging, and bioinformatics. And she was the first author on the Frontiers in Science lead article on the model that we're going to talk about today. Jay Coggan. Jay is the founder and president of Neurolinx, a non-profit research institute dedicated to advancing neuroscience research through open scientific collaboration and data sharing. Previously, he was senior scientist in the molecular systems group in the simulation neuroscience division at the Blue Brain Project too. Jay's research is focused on synaptic plasticity mechanisms, ion channel fluctuations, cell metabolism, and models of demyelination diseases. Polina, Jay, thanks very much for joining us today, and it's over to you.
So we studied this neuron glia vasculature system and the metabolism and the electrical signaling in this complex ensemble of the cells. And this was an amazing, almost 6-year effort that is, I would not believe in the beginning that we will achieve that much, that we modeled the 183 biological processes with the 95 enzymatic reactions and 19 transport processes, and also like 69 other processes related to the ion currents, blood flow dynamics, and some non-enzymatic processes, like, for example, related to magnesium binding. And we have the 176 metabolites in the model, and the model itself is 151 differential equations. And what you see on the right in this slide is actually the model. Like every single piece is what can be studied with it. So if we go next, how did we build this model? So it, of course, started from the data collection that was immense journey to get it from all the publications and the databases. And then I was building the single models of individual enzymes or transport processes, and then iteratively refining them, then combining them into groups of processes, into pathways, and then further refining, validating the model, growing it up to the neuron and astrocyte models, and then the entire NGV model. So that was really like almost 6 years of iterative refinement of the model, like optimization, validation, all these stages. So this could then tell us how we can simulate the healthy state, brain energy metabolism and signaling. But then Professor Henry Markram told us to add the aging in this model. And I was first like a bit skeptical, like it's going to break. But to my greatest surprise, it actually worked. So I incorporated the data from the RNA expression study that was publicly available data on the changes of the levels of enzymes and transporters in neuron and astrocyte, and also the data, it's a bit slow, also the data on the blood nutrients availability, synaptic glutamate release changes, and NAD and ADH pool, and the NAD and ADH cytosol mitochondria shuttle, which is like a generalized expression of some set of reactions. So then we could learn many things, and Jay will start with some interesting findings.
Thank you, Polina. So how do we go about studying the effects of aging on neuronal signaling? Well, we looked at the effects of aging on action potentials or voltage spikes. It is generally well known that the pattern of spiking conveys information and that spikes are energetically costly. But not all spikes are the same. Individual spikes contain information about neural activity and also need to be optimized. For example, the amplitude, the rates of rise and fall, the width, and the long tail called the after hyperpolarization, or AHP, all contribute to the costs of spiking. These features are changed in the aging state and can reflect energetic dysfunction. Aging also affects metabolite levels in both neurons and astrocytes. Sometimes these levels are elevated and sometimes depressed, indicating the need for critical balances in proper metabolic function. Of particular interest here, we can see how levels in the energy-carrying molecules ATP and NADH are reduced, no pun intended, in aging in both neurons and astrocytes in most compartments. These changes could also affect synaptic activity and many other things. So when we arrange metabolites according to metabolic groups or pathways that they belong to, certain patterns can emerge. For example, here we can see that during activation and aging, the levels of the excitatory neurotransmitter glutamate, as well as metabolites from the TCA or Krebs cycle, shown in light green and dark green, are even more affected than those in other impaired groups. These changes can also affect synaptic activation, as well as almost every other brain function. So overall, we see a trend toward lower supply and demand for energy in the aged brain. For example, in panel A, we looked at the adenylate energy charge, which is a measure of the ratio of energy-rich ATP to energy-poor ADP and AMP. This indicates the cell's energy status. A high AEC suggests sufficient energy for all cell functions, including neurotransmitter synthesis and ion pumping. The AEC is lower in aging for both neurons and astrocytes. In panel B, we see that the ATP costs of running the sodium-potassium pump differs for both neurons and astrocytes in aged brains. In the old group, the ATP usage in neurons goes down, whereas consumption goes up in the astrocytes. The sodium-potassium pump is critical for maintaining ionic equilibria and many other cell processes. And then finally, in panel C, we see that the ratio of astrocyte to neuron sodium-potassium pump activity is elevated, suggesting that the neurons are less active compared to the astrocytes in the aging brain. This finding suggests that there is an increased demand on astrocytes to support neuronal function, such as clearing the extracellular potassium in order to maintain correct Nernst potentials for spike generation, and many, many other cell functions that can affect overall brain function. And then back to you, Polina.
Yeah, so we then thought, what else can we learn from this model in which we can control like every single element, as in the biological experiments, we often cannot afford such flexibility. In computational models, we do. So we looked at the effects of potential like molecular damage in these cells, which is in a sense a sensitivity analysis where we perturb one parameter at a time, and this results in a suboptimal function of corresponding enzymes and transporters. So in the figure A, you can see the typical trace for the ATP in the neuron that is at the standard model, let's say with 1P, or with this P parameter of some enzyme being increased or decreased, and how it affects this metabolite and all the other metabolites in the model. So we could also look at these effects, the sensitivity changes at the rest state and when there is a stimulus given to the neurons. So we could see that actually they are different, the sensitivities of the rest state system and the stimulated system. And then they thought like this resembles like how would the cell or the ensemble of cells respond to the stimulus in this kind of damaged state. So they then thought like probably it can be different between old and young systems. And indeed, that was really interesting and surprising to see that actually neuron and astrocytes were the opposite in this metabolic adaptability, which suggested that there is some intercellular communication that is probably changing with aging. They then were also interested to more formally and quantitatively evaluate these sensitivity or adaptability networks, where we have all the metabolites and we have all the enzymes, all the kinetic parameters. We connect them by edges that are weighted reciprocal to this metabolic adaptability, and we build these networks as you see in the bottom of this slide. So we could see that the aging brain is more fragile and susceptible to damage, as we quantified by the node centrality in this adaptability network. So how far we go from one node to reach all other nodes in the networks, basically. And then we also looked at if we cut these edges like by percentile like, of how long they are, what will be the system look like? And we looked at the number of connected components in such filtered networks, and surprisingly, the old network was breaking into these disconnected components faster. So we got these kind of connected islands that are separated in the old network faster than in young network in this process of filtering. And then further, our amazing colleague Lida Canary performed the algebraic topology analysis on the same networks, and I was like super excited to see her results also confirmed our findings, that from the point of view of the topology, also the old system was more fragile and the young system was more resilient, as you see in the bottom in the various complex characteristics of this network. And then we go further, we could look at these edges, these connections between metabolites and enzymes, as potential drug targets, or the enzymes themselves, which possess these most vulnerable, most fragile links. So we then selected these enzymes and performed the transcription factor enrichment analysis, because the ideal kind of drug target would be some factor, some molecule that would act on this and bring our complex, very interconnected system to the young state. So we found that ESRA transcription factor was the top scored in this analysis, and that was surprisingly linked to lots of mitochondrial and aging-related processes in the independent literature. Sorry, I got a bit cold. We could then look how the system performs if we target to bring it to the young state from the old state. So we selected these most vulnerable targets and the related parameters, and performed a constraint parameter optimization. But additionally, we also selected the parameters that are related to such lifestyle interventions like a diet, exercise, and also the supplements that were known from the literature, like NAD. Sorry again. To our surprise, this constraint optimization resulted in the diet, exercise, NAD therapy, which was very close to the top targeted therapy, also as a result of the other constraint parameter optimization run. The difference is only which parameters we set up for the optimization. And what was great to see that the optimization converged to that known lifestyle of lowering glucose, having exercise, which is reflected in the lactate level in the blood, by the way, and this NAD supplementation. And we could see the effects on the metabolite levels, but to bring the effects to the electrophysiology, to the firing of the neurons, we also had to reverse the aging effects of the aging decrease of the sodium-potassium pump. And then with these rejuvenated systems, we evaluated also the same metric of the network fragility or adaptability, and we could see that they actually improved and were even better than those of the young system. So our old metabolism was repaired and even got a bit better. And with this, I'd like to give a word to Dan to show how you can use this model and also model other diseases, for example.
Thank you very much, Polina, for that deep dive into this new research model. And a quick reminder, if you have any questions you'd like to ask the panel, please keep them coming in. We have questions already, and we're standing by to ask as many of those as we can in the time we have later on. But before we do, our final presentation is Daniel Keller. Daniel Keller is Research and Development Scientist at the Open Brain Institute in Switzerland. Previously, he was Group Leader in Molecular Systems at the Blue Brain Project at the EPFL, where his team constructed and ran advanced mouse brain models on a supercomputer infrastructure, enabling unprecedented in silico experimentation and hypothesis testing. Now, at the Open Brain Institute, he's helping develop new capabilities for large-scale brain simulation. So, welcome, Daniel, over to you.
So the Metabolism Notebook can be run at the Open Brain Institute, which is charged with making the models developed at the Blue Brain Project accessible to the entire world. So at the Blue Brain Project, we did develop this large-scale model of the mouse brain, which we simulated in a supercomputer, and we're exposing that. You can explore whole brain models at the Open Brain Institute, see the different morphologies and compositions of all the different brain regions. And we're working to add the human brain as well. So you can create virtual labs to build and run simulations of the neurons and neural circuits in the brain. So, okay, so how do we actually access this model? Well, you go to openbraininstitute.org, click on resources, and the Brain Metabolism Notebook is the first model in the showcase. And so, showcase, there we are, it's a little bit slow. And when you click on it, it'll open up a Jupyter Notebook. The first thing you have to do is just sign in with a GitHub account, and then it'll take you to a Jupyter Notebook run from the Institute servers. In this notebook, you can simulate the metabolic responses to stimulation step pulses as described in the paper. So the model first reads the initial concentration values and parameters, and you can then modify them as desired. There's hundreds of different numbers that go into this model. You can then configure the stimulus and age properties, as well as the stimulus onset and the total time of stimulation. The most important parameter, of course, is the age, which can be set to either young or old in a pull-down menu. A variety of additional configuration parameters can also be set. The simulation also supports custom mode, in which parameters can be modified to support simulation of particular disease states. So the equations are defined and solved using the Julia solver. You can select which concentrations that you then want to plot in a series of checkboxes. And so there's hundreds of different potential items, and you wouldn't want to look at them all just to avoid being overloaded. And then you can plot them and perform subsequent analysis. And so that's essentially an overview of the notebook. I urge you to check out the other notebooks also available at the Open Brain Institute, as well as explore other capabilities we have in it. We're adding new stuff daily, so it's a really exciting time for the Open Brain Institute. Thank you for your attention.
All right, thanks very much, Dan. So it's time to move to the panel discussion. In addition to, we have questions for all our speakers: Henry Markram, Polina Shichkova, Coggan, Daniel Keller. But also, I'm delighted to invite two additional guests to join us as well. Firstly, Professor Mark Mattson, Adjunct Professor of Neuroscience at the Johns Hopkins University School of Medicine in the US. Mark's one of the world's most highly cited neuroscientists, best known for his pioneering research on brain aging, synaptic plasticity, and neurodegenerative disorders. As chief of the Neuroscience Research Lab at the National Institute on Aging, Mark led research that uncovered mechanisms by which intermittent fasting and exercise enhance cognitive function and protect the aging brain. He's author of influential books, The Intermittent Fasting Revolution and Sculptor and Destroyer, and as many of you may know, host of the popular podcast, Brain Ponderings. Our second guest is Pierre Magistretti, Distinguished Professor and Vice President for Research at the King Abdullah University of Science and Technology, that's KAUST, in Saudi Arabia. Pierre is renowned for his research on brain energy metabolism and glial cell biology in particular. His laboratory proposed the astrocyte neurolactate shuttle model, revealing how astrocyte-derived lactate supports neuronal activity, memory, and neuroprotection. These insights have been fundamental for interpreting functional brain imaging and understanding neuroplasticity. Pierre is past president of both the Federation of European Neuroscience Societies and the International Brain Research Organization. So welcome, Mark. Welcome, Pierre. Thank you very much for joining us today. So to begin with, we have many questions, and we'll come to as many as we can, as I say, and perhaps we'll try and group some of them together, particularly focusing on ones which go beyond the article itself. Please do visit the article online, where you can, some of the questions will be answered as well. But to begin with, Polina, the model that you showed, a particular finding was that age-related changes in metabolism impaired the generation of action potentials by neurons. Could you tell us a little bit more about the mechanism that's causing this, particularly, I believe, the sodium-potassium pump?
Yeah, sure. So that was really interesting to see that while our changes are localized to the metabolism part, we see the effects in the action potential generation. And as you mentioned, as we describe in the paper, that is mostly attributed to the changes in the sodium-potassium pump, its concentration. So in the model, we tested different, so this pump has regulatory subunit and those that are doing work. So you need to bring these differences to these subunits and then see how the pump is affected. And there is also such component as the ATP level, which contributes as well to these differences, but that was to the lesser extent as we tested. And yeah, the effects we observed were on the shape of action potential, its amplitude, all other firing characteristics that Jay introduced and also available in the paper in supplementary even more. Yeah, would this answer your question?
Yes, thanks, Polina.
Perhaps I could just add on to it. Some of the changes also enter in through the ion concentrations, which cause changes in the reversal potential of different ionic species, which in turn can affect the shape of action potentials.
Thank you very much. Pierre, how do you interpret that? Can I yourself and particularly yeah, actually let's move on. Mark, I think we've just lost Mark Mattson briefly. Perhaps if I could just switch before we come back to Mark on that. Very briefly, Jay, you found certain findings with respect to the astrocyte to neuron lactate shuttle. Can you elaborate on what you found, what the model showed, what was novel, what was surprising from that? And then perhaps we'll ask Pierre to comment on those findings himself, having proposed that model some years ago. Jay, over to you.
Yes, well, normally the astrocytes will, in fact, in the simulation in the young group, the normal case was observed where the astrocytes provide lactate to the neurons for activity. And then the neurons take the lactate in and put it through oxidative metabolism. But in the old group, what we found was that the flow of lactate wasn't going always in the correct direction, and that lactate levels in the astrocyte, the intracellular space, the neuron, were not exactly what they should be. And as I said in the data presentation, that the concentrations of metabolites are absolutely critical to the normal flow of metabolic function and energy production. So if something is going wrong in the neuron, sometimes, for example, we saw that this TCA cycle was not working well in the neuron. That could cause pyruvate to build up, which would then cause more lactate to be produced in the neuron, which paradoxically we saw then being exported into the extracellular space, which is not really supposed to happen in a normal function. At the same time, we saw the astrocyte struggling to keep up and provide more energy to the neuron. And it did that by producing more lactate. So we had sort of like a traffic jam of lactate going on.
Right. Pierre, you proposed the ANLS some years ago. How are the findings of the predictions and the findings of the model, how do they relate to the latest understanding of how that model operates and how it's related even to the neurodegenerative disease in aging?
Yes, I mean, I guess the modeling in the young is, confirms the experimental data, which is always nice. In fact, as Henry reminded us, we did some, let's call it rudimentary. For us at the time, it was quite elaborate, but of course, with the technology and power and knowledge that the Blue Brain project has accumulated, that was pushed to highest levels, like we see in this paper. But with Renaud Jolivet, we actually had models and confirmed the experimental data in the young. Now in the old, this is very interesting. I think this finding. I like what Jay mentioned. and that is the term like a traffic jam between astrocytes and neurons in terms of lactate traffic. I think that the hypofunction of the TCA cycle in neurons obviously is certainly a component in this. I just have a question actually for all Polina, Jay, and Daniel. The experimental data in which you tested, and correct me if I'm wrong, in which you yes, modeled the ANLS was by increasing the core of the data are increasing glucose, circulating glucose, and scaling the increasing glucose with lactate. And this is the input function, if I may simplify, to the system, and you have all the equations, the partial differential equations on all with transporter enzymes and all that. I was just wondering two things. First, does the idea of scaling lactate in the arterial lactate with glucose, I wonder how required that is, because there can be physiological conditions or pathological conditions also where glucose is high, but there is not necessarily a commensurate increase in lactate. And that, or just take exercise, it will modify the lactate concentration. So all I'm saying here is, did you try to also run the model without scaling lactate? Maybe it's a bit detailed, but I think it has implications. We'll get to that in a second. Because the second point is, okay, you increase or modulate, which is absolutely fine, the input function. And let's go now only for glucose. In principle, at least in the young, you have a kind of a linear relationship, more or less, within a certain range between arterial glucose and then intraparenchymal glucose, more or less. I don't know exactly the function, I'm sure you know. But what may happen is really, and that's actually documented, that there may be decreases in the ability of the brain to import glucose with aging. In other words, for example, decreased GLUT1 expression that has been documented. So you can modulate the glucose in the blood, but then what is important is how much gets in aging, because the transport is somehow affected by aging to the point, and I put many quotation marks, but there is this idea that Alzheimer's could be a sort of type 3 diabetes, a diabetes of, in other words, that like muscle in type 2 diabetes, unable to bring in glucose, that may be something that happens in aging. So just to wonder if you kind of took all these factors into consideration.
Yeah, so may I answer these amazing questions? So first of all, we do have the scaling of the glucose transporters, and that is the cell type specific based on the single cell RNA-seq data, and it's different for astrocytes and neurons, and also for the transporters of the blood vessels. And that is fixed aging modification of the system. Then for testing the lactate, we have actually both setups, and they are described in the final published Frontier's paper. Initially, we had only this scaled version, as you described, and that was on bioRxiv, but then we also included where we have no link between lactate and glucose. So that the results, they are a bit different, but we really described it in details, and we provided both figures in the supplementary, the Frontiers. And that was actually like really thanks to our amazing reviewers, their input, their suggestion that we should look more into this. And this was one of the latest additions, like exactly as
I'm not surprised, as both Luc and Jolivet were postdocs in my lab.
Yeah, so also like as you point, like this scaling, this interdependence, it's really like we don't have enough experimental data to really judge this, which of these models is the correct one. So we provide both, and then it's up to the wet lab experiment, I would
So
say.
you have to look, so you have data of the modeling with when you don't scale lactate.
Yeah.
Yeah, I haven't, I missed it, but you said it's in the supplementary.
Yes.
That's a good lesson for me. I
It's in
should
the
also
latest
read the
version in
supplementary
Frontiers.
data.
Yeah. We had only one in bioarchive, and then we have more on Frontiers.
No, I read the final paper, but I didn't read the supplementary.
Yeah, it's in the supplementary in the PDF with the with all figures. There you have to
We've had a few questions in actually on aspects such as validation, how the models are validated. I'll perhaps leave some of those because it's extensively described in the article as well. But moving on, I think Mark is back with us. Hello, Mark. Mark, we were just talking before, just to bring you back in on a question that came before. We were talking about the findings of the model on age-related changes in metabolism affecting neuronal activation and excitatory activity. And you wrote the editorial that accompanies and comments on the model and its findings. And this is one of the things that you flagged and drew out yourself. Could I ask you from your perspective, how do you see this relationship between metabolism and neuronal excitation, and it's linked with neurodegenerative disease? What contribution is excitation bringing to the onset of disease?
Well, one thing I could point out is these the sodium pump, the glucose transporters, they're in the membrane, right? In the outer membrane of cells, and they're in a lipid-rich environment. And it turns out that those two proteins, this is an example, the glucose transporter in neurons, GLUT3, and sodium potassium ATPase, they're vulnerable to being impaired by lipid peroxidation, so oxidative attack on membrane lipids. And so you could imagine that, and that's one hallmark of aging, oxidative damage, among probably a dozen other common things: impaired energy metabolism, impaired autophagy, et cetera, et cetera. The question in all of this is, and that we think about, but maybe we shouldn't so much, is like, what comes first? And I kind of view aging as a stochastic process, and it's not necessarily going to be one particular vulnerable pathway or protein, and it may vary between individuals, but once one pathway becomes tweaked out of whack a little bit, then that's going to impinge upon the other pathways that contribute to aging and dysfunction. Another thing I want to point out is that organisms without vascular systems and neurovascular coupling, their brains age, like worms and flies, for example. Well, flies kind of have a vascular system, but so if a goal is to understand like common mechanisms of brain aging and decline in function with brain aging, then one wonders whether, should we be looking for explanations that are completely different than mechanisms in other organisms? So that's something I throw out there. We didn't talk about it all yet, but it's an important point. And I think this estrogen-related receptor is an interesting thing there. Are there homologs in worms? There's some good. Gary Ruvkin had a postdoc, Kathy Wolkow, who ended up coming to my lab at NIH, and she'd shown in his lab that impaired insulin or altered insulin signaling, insulin-like signaling, in just a few neurons in the brain affected aging of the entire organism. So I think there's a lot of interesting things related there. And then finally, I don't think ketones have been mentioned yet. Of course, with Pierre, talks a lot about lactate, but there is evidence that during brain aging, whereas glucose uptake ability of neurons may be impaired, ketone uptake, beta-hydroxybutyrate acetoacetate, may be less impaired or even early on not impaired at all. And I guess we're going to talk about lifestyle factors, but the only three things we know to slow down brain aging and promote healthy brain aging are physical exercise, intermittent fasting / caloric restriction, and mental activity in neurons. And all of those, well, the first two can involve metabolic switching to ketones, but they all involve imposing a mild intermittent stress on the cells and then and triggering adaptive stress responses that protect against aging. So I know I threw out a lot there, but maybe the listeners, of which there's hundreds now, may have some comments there.
Yeah, I think we'll come to interventions shortly. Before we do, could we move? One of the major findings of the article, as Polina described, was the finding that the aged brain was fragmented, the metabolic system was clustered, if you like, as compared to a more even distribution. Oh, I was going to ask Henry. Henry, I was going to Henry, I was just Mark just brought into play the point about the connectome, if you like, the rather than looking at single mechanisms, the nature of the behavior of a network. And of course, this is what we have here. We have a network analysis that showed the fragmentation of brain metabolism. Can you just develop that thought to us? How important, what's the implications of taking a network perspective to the question here, and what does that tell us about what we need to be researching further?
Well, maybe just before that, just to address a point that Mark made, which is that there are organisms like worms that have no effective vascular system and their brains age. What we may consider is that eventually you need a vascular system for more complex brains and then you would need this complex metabolic system in order to support all these neurons. If you look at the vast volume, I mean, your brain, if you unwrap your axons, it's enough to lasso the moon. They need a lot of support. So I do think that the evolution brought into play a very, very sophisticated metabolic system in order to support the neurons. And while they are clearly, I mean, we know aging occurs not only in the brain, it occurs in every cell in the body. I think that the energy supply, or making sure that you can support all facets of neuronal function, synaptic transmission and activity and so on, it would be absolutely critical. And if you do simulations and you lose even a tiny bit of your action potentials, you get significant changes in the ability of neural networks to generate activity patterns. So, but in terms of the network, what we found, so we're very network-oriented people, and this is a network of 16,800 different pathways. So one shouldn't think of the metabolic system as being an enzyme or transporter or anything else. It's really a massive network, and it's a very sophisticated network, so that if you have a certain demand, cognitive demand or activity demand, then the network can respond to provide the right profile of metabolites in order to be able to deal with that and support that cognitive process. And then if you switch to another cognitive process, the network is capable of responding. It's also much more resilient, so that if you get a little, perturbation on one end, it's kind of absorbed by the network. And if you have a very distributed network the way it is in the young brain, it's absorbed, which in a sense that it doesn't completely go out of range. So it's like you can support the metabolism under many different conditions, and in many different environments and stimulations. And so you can think of it as sort of a cognitive repertoire. You can support a wide cognitive repertoire. When you see the network getting fragmented like that and becomes sensitive to perturbations, a small perturbation in one of the metabolites or transporters or ion channels, or in a certain compartment, and you see that there's a tiny perturbation there that actually leads to a huge effect in other parts of the network. This is not good for stability and robustness. And so what we can imagine happens is that you basically are reducing, by reducing adaptability of the network, you would be reducing the range of activity profiles that neurons can undergo. And by extension, you would limit the cognitive repertoire. So in a way, it's a bit like the body, physical body. If you keep, you do yoga or something, you keep your repertoire huge. But if you don't do exercise and you don't do any of these, your repertoire shrinks, and eventually you can only walk in a certain way. And I think that probably this loss of metabolic flexibility actually also translates into a loss of cognitive flexibility.
Thank you. And so moving on to, we've had quite a lot of questions about applications, if you like, toward drug development, toward interventions. And so what you did in the study is to use that network analysis to prioritize potential drug targets according to their ability to improve the most pathways. And estrogen receptor alpha, ESRA, came out, the transcription factors having the best score, if you like. It was the best scoring one, and then you identified relevant proteins associated with that. So Pierre, could I ask you to comment? How do you view the potential of ESRA, and Mark as well, please come in. What do you, both the potential of the methodology here, its utility in predicting future and future of the drug potential targets, and specifically ESRA, how does that align with other evidence?
Yes, I mean, this could be a very useful, and I think that's that it was thought through for this application, among other things, to identify potential targets that would normalize the metabolic coupling or metabolic disruptions of the coupling that you have described. So given that you have so many targets, transporters, enzymes, transcription factors, epigenetic markers, I mean, a huge array of potential targets, and that out there, there are many agonists or antagonists of these various targets. It would really be, I think, very exciting to try to model. I mean, if I increase this enzyme, or I increase this transporter, or decrease, or even upstream on a transcription factor that controls a number of genes, do I have what happens to the model? And what happens to the network at all the levels, metabolic, in terms of metabolites, but then eventually on the network. And so, and I can personally see, by the way, I may, and I actually thought for a while to getting in touch with you, because we meet, I mean, I am back in Geneva and Lausanne once in a while, to based on the work we're doing, with the spin-off from my lab at EPFL, Glia Farm, molecules that we know have specific targets to enhance, in particular, astrocytic metabolism, and see in this model what would be the consequence. I really can't wait, in a way, to. We have a lot of experimental data, but it's always nice, again, if we can explore this also from the modeling point of view. So definitely very useful tool. If I understand correctly, it's possibly more what you are doing, Daniel, with the company. I mean, the open, not the company, but the software, Julia. I mean, you showed some people can have access. I mean, it would be nice to collaborate, but I mean, I understand that it could also be accessed. It's an open source, correct?
Yes, exactly. Actually, conceivably anyone can build upon the models, already present. And I think the great thing about the Open Brain Institute is that it will allow many people from different institutions to have a platform to collaborate together.
Yes, I think it's fantastic. This is really a very significant contribution.
So maybe just a quick comment. So in the you can create a virtual lab, and then you can invite lots of colleagues, and so together you can decide what the project is. It could be on, like trying out a drug which you think would alter one of the metabolites or transporters, and then you could explore the effects. Or it could be on whatever hypothesis you have. So it's really for hypothesis testing. And of course, this is only the beginning. We're going to be advancing this model more and more. We're also moving to being able to add this into the geometry of the neurons in different parts of the brain. So you could then zoom in and do this model in any part of the brain, so that you can see whether there are differences in different parts of the brain. So I think and together, every time, the more users use it, the more people are going to identify there's something that's missing or something can be improved. So this model will sort of become a community collaborative effort to become even more advanced. And it's completely open. I mean, it can be downloaded and run on your own, but it's much more effective in a virtual lab where you can actually build on it and others can build on that as well.
I agree. Fantastic.
Yeah, Mark, coming to you, there's quite a few questions in the chat about the application of this to supplementation, dietary supplementation, in particular with NAD. The article showed that found that the combination of diet and exercise and NAD had an effect similar to the sort of top 10 drug target. From your perspective, how does this model align with the latest we know about lifestyle intervention, exercise, and dietary and NAD supplementation? And again, how do you foresee the model being used in that respect?
That fits quite well, in my view. So these different interventions that are good for the brain in general and may mitigate changes with aging, whether it's in energy metabolism or, functional properties of the networks. Again, they all enhance the ability of the cells to cope with stress, whether it's oxidative, metabolic, and so on. And how do they do this? For example, stimulating mitochondrial biogenesis, upregulating antioxidant defenses, promoting autophagy, etc. Right. So I think kind of the key thing, though, is the modeling is really great in identifying, in this case, a potential target, the estrogen receptor alpha. And then what needs to be done is to go back to an animal model and manipulate that gene up or down, either globally or only in astrocytes, only in neurons, only in whatever, vascular endothelial cells, and then see how that affects age-related, I guess, cognition decline, or a lot of times you can you can see a bigger effect of some manipulation when you challenge a system with some severe stress like metabolic stress, ischemia, or seizures and so on. So I think that's the way to go. The modeling can provide a lot of new information, but in my opinion, you got to go back and actually do experiments to validate that the modeling is, you know, that it's leading you in the right direction.
Okay, we're coming to the last sort of quarter of an hour or so. I'm going to go more specifically to a few questions that are in the from the audience. And first of all, it's methods. It's back, Polina, to you, and Jay to you, I think, and maybe Dan. We've had several questions grouping about various methodological sort of limitations in the data, and what do we know about sort of applications. So to group a few together, what about the? Do you plan to introduce proteomics into the data? Where were there limits on what you could include, what you could model? What were the outstanding limits, if you like, and things that were not possible to do and perhaps might be possible in the future? Also, human versus animal application, if you like. How confident are we that if most of the data were from the mouse brain, that this would apply to humans? I guess speaking to the limits on human data. And lastly, gender differences. Are we aware of any gender differences that might impact the findings of the model? So I don't know, Polina, Jay, if you could handle some of those. Proteomics, human animal, male, female, and missing data. What were the limitations of the modeling process?
So yeah, of course, like as any model, it has lots of limitations, but it offers lots of new findings as well. And we described all these limitations in the dedicated section towards and like after the findings, after the discussion, I think, or in the discussion, don't remember exact location, but there is a dedicated section on the limitation where we were transparent. We try to be as transparent as we can. And of course, there is this limitation in the available data is the main reason why we would model the rodent and not the human, because human would be like super interesting and relevant, of course. But as more human data becomes available, this model can be easily humanized, so to say. Like when we bring the human data, we can use the same or similar equation in most cases, but for the human system. And then the sex differences. So unfortunately, it comes again to the limitations in the data and the data availability. And clearly, like especially we find in the estrogen-related receptor that calls to the question of the sex differences. We did not implement that, but I'm really looking forward to see how people will incorporate such data who are more experts in this aspect. Yeah, that would be really interesting to see these two different models and how differently they will age.
Just to add something to that, I think, what is really important, and it's also related to what Mark said, a model is not, a model is for hypothesis testing, it's for exploring. You got to go back to the experiment. But one of the things I'm an experimentalist. One of the things I find very valuable in a model like this is it provides a framework to show what kind of data you need. So for example, if you look at the model and you see what is the data that it needs, it becomes much easier to go into many other studies, different brain diseases, gender studies, age during development, and you know exactly what to measure. What we found in the 20 years is that scientists tend to measure what they are sort of married to. They measure things that they like because they were introduced to it in a certain lab, but they don't necessarily measure all the things that are needed to be able to build a model. And so the model serves as a very important reference of what it is that you need to measure if you want to study epilepsy, or if you want to study some other disorder and changes. If you do only half of the data set that is required by the model, you're not going to be able to make these kind of insights. So I think that it is an important guide to how one should go about doing the experiments.
Thanks, Henry. Speaking to the future use of the model, Dan, if we can come to you, there's a few questions with respect to things like how the model can be used in the future, looking forwards, if you like, and can you speak perhaps to its further refinement on the platform, and aspects such as what kind of collaborations are needed to move toward to develop it to make it even more clinically relevant, if you like. What its applicability, we've been asked, in Parkinson's disease and other conditions as well. Does it have particular applications in other conditions? We've spoken briefly about dementia, I think specifically, but other conditions such as Parkinson's. And also, we've also had questions about its relationship with inflammatory processes within the brain as well. Is the relationship between inflammation and metabolism something that could be built into the model?
Okay, yeah. So at the Open Brain Institute, we're starting to run simulations of neurons with defined electrophysiology types, morphology types, and soon transcriptomic types. So this will give the community the capability to determine the energy needs and susceptibility of particular types of cells, and allowing you more detail. So you mentioned, several particular diseases or conditions like Parkinson's and inflammatory conditions. So I think all of those can actually be modeled by extending the model that Polina developed. And if you need more detail in a particular pathway, you can just add detail to that pathway and still reuse the entirety of the rest of the model. But there's also a large number of future directions that the platform will allow people to do. For example, I saw some of the comments were about MRI and blood flow. And so we've developed a model of blood flow that can run in the vasculature superimposed on the brain, and it's modulated by the neuron and astrocytic activity. And so I think that will actually also be super useful for studying some of these conditions.
Yeah, thanks. If we just last sort of 10 minutes or so, zooming out, the hub in which the article is published also has a policy outlook by Renaud Jolivet and colleagues in Maastricht, and they point to the need for an understanding of a fundamental understanding of the importance of investing in the kind of research we've talked about here, computational neuroscience, and therefore the need for sustained investment over long periods of time and together with the kind of infrastructures that are necessary. And I wonder, Pierre, Mark, if you could comment on that from your own perspectives, obviously in different parts of the world. Would you echo the lack of awareness of the need for this kind of investment? What should we be telling decision makers who decide on research funding, be they private, be they public, on where we should be focusing research efforts in order to sort of build the sort of capacities that Henry and team have described here?
Well, there's so many reasons we want to understand how the brain works and what can go wrong, how to make it function optimally. Most of the money, so this modeling is fantastic, and it relied on tons, lots and lots of data from probably hundreds and thousands of labs and integrating it. And so the funding has to be at all levels, and the input, there's still, as I mentioned, there's still a lot of need for more information, right? For example, we still need to have spatial resolution of some of these pathways and so on, right? Rather than more of a global changes with age, in this example of the aging. Yeah, there's a huge need. The United States, we're in a mess right now because there's really, I don't understand it, but kind of a dishing of everything we've accomplished, or the approach we've taken in this country to develop basic research and build up from the ground. So I think that that's the key. The continued funding is necessary. And for example, the NIH now, they're apparently going to greatly reduce or maybe even eliminate using animals, using vertebrate animals, which is crazy. So I would say that funding is essential and important if we want to improve human flourishing in the future.
Absolutely.
Yeah.
And likewise, Pierre, your perspective before, and then I'm perhaps going to come to all of you, including Jay. I'll give perhaps a final word in a moment on this. But Pierre, just first of all, respond to that. I mean, where you are.
On this one, obviously, funding is important. I mean, I am lucky to be in a place where we have one of the most powerful computers in a university, Shaheen 3. Which is used very massively, for example, for measuring for climate change, predictions, for building digital twins of coral that are suffering and interventions. And so you can, what I'm saying here is two examples that are very concrete, that mobilize a considerable computational research. And of course, the brain would be another ideal. And as Henry mentioned, we collaborated on this. And of course, Henry and Patrick Hebischer in the early 2000s were visionaries in starting this at TPFL, and I was lucky to be part of this when I was there. But so I think there is no doubt that you need this, and if you add another layer, which is the AI. I mean, we hear a lot about this, but this is not just a buzzword now. It's a reality, and for that you need also important computational power. So yes, investment there. Of course, experimental and clinical work should continue and develop even more, but definitely modeling computational is very, very important.
Jay, can I ask you to comment? Last comments actually from everyone. So Jay, Neurolinx, of course, by definition, it's a network. It's computational neuroscience. So what is your perspective there, and specifically how do you see computational modeling moving forward in the next 10 years?
Well, I think computational modeling is incredibly important, but it doesn't stand alone. It's a partner to laboratory biology. I started my career as an experimentalist with real animals, and I made a transition to be a computational biologist. So I understand both worlds very well, and they're both incredibly important, and you can't have one without the other. And there's a lot that happens, especially if you want, to study diet and things like that. There's a lot that happens from plate to brain, if I can introduce that phrase. Like there's something called bioavailability. There's a lot of biotransformation that occurs from what you eat to what actually winds up in your brain. Controlling that is very difficult. You need to do animal experiments, and the computation we can magically insert the right concentrations of things like ATP or NADH here and there, and we can say theoretically this is what should happen, but then you got to develop the drug or the diet or some sort of regimen to actually make that happen, and that's much more complicated. And so you need both. You need both laboratory and computational experiments.
Yeah, thank you very much. Okay, so just last statements, really. I'll perhaps go Polina and then Daniel and then end with Henry. Polina, thank you for bringing this article to Frontiers in Science. Look forward to your further work. And from your perspective, having developed this, where would you like to see the model be developed next?
Yeah, so
Used next, I should say.
yeah, thanks for the great question and for like everything, this amazing discussion with the experts. And really, I really hope that the Open Brain Institute will make it a big collaboration effort, that other people can further bring the new pathways that we didn't cover, because that's the PhD time is limited. So I really hope that this model will further be expanded and refined. And that's now I'm working in some that different field, but I'm happy to help also. And if your question is unanswered, or if you're watching it later, you can contact me on LinkedIn or by email or on the GitHub, and then I will try to help you. Like, yeah. And I'm really looking forward to see how the people will use this model. And also to comment a few seconds, we already applied it to some disorder with the J and others, and the paper is published as well, published just recently. And also we applied it in the circuit of cells, and that is coming soon as well.
Great, thank you. And Daniel, the future of OBI for yourself?
Yeah. Well, so I would kind of echo, Polina's sentiments. Throughout my career, I've seen graduate students and postdocs, and they come in and they put in a Herculean effort for a few years, and it results, in seminal publications. But then, in other places, the next grad student comes in and they have to start from nearly, zero and, work their way up the same slope. It's my hope that through collaborations like and initiatives like the Open Brain Project and others, that people will be able to build upon what's been done so that, we can expand our knowledge together.
And lastly, Henry, if I could ask you to close out with you brought this project to Frontiers in Science. You're leading the Open Brain Institute. What's your vision for this organization over the next 5 to 10 years?
Well, I think it's as Dan says, is so we can really build on each other's shoulders. I mean, that's what the project, that's what, when you build these kind of models, you're really building on the shoulders of giants. And we think that everybody could be building, and we could go much faster if we actually built on each other's shoulders. One of the most important things, I just share with you, one of the most important things we discovered in attempting to build the brain, the first thing you come across is that there's not enough data, right? And you haven't measured everything. The number of pathways just in a tiny piece of the brain is less than 1% of the data is available. But that's the problem that we actually had to solve. And the fact is, you don't have to measure everything. You can infer a lot. And it is really, especially in the time when you're under pressure for grants, that you need to measure something or you need to do a certain experiment, the value of these kind of models is that it can help you really build a very strong case for measuring certain things. We identified lots and lots of data points that are absolutely critical. You can't build, you can't continue if you don't measure, and whether it's a phenomena or whether it's a concentration of a protein, but I can tell you that 99% of the data in the brain does not have to be measured. You can infer it in the process of forward engineering. So my favorite saying is, you can only reverse engineer the brain if you forward engineer it, and you can only forward engineer it if you reverse engineer it at the same time. So it's a loop. And I think that what I hope for the next 5-10 years of the Obi Kenobi, is that we have Jedi scientists that are able to move at lightning speed. In fact, what we've done is we put an AI on top of the 18 million lines of code, on top of everything. And so you don't have to be a computational neuroscientist to build these very sophisticated models. The AI can help you build neurons, synapses, circuits, hippocampus, whole brain regions. So now we've made it as accessible as possible for almost any student of neuroscience to go ahead and to build models. And I think that as you do that, you sort of bump into the data and say, oh my God, I got to go back to the lab and I got to measure something. That's great. That's a discovery. That means that that is really critical data. But we have to be careful because there is a lot of data that can be predicted. And you don't want to measure stuff that can be predicted. You want to measure stuff that can't be predicted. And it is a small fraction. So one does have to be very careful as we go forward. But it's also good news because it means we can go so much faster and we need to collaborate. And so the last part is that, you know, I see a sort of global worldwide collaboration. We already have close to 200 virtual labs operating today after a month or two of opening. We opened in January. Hopefully there are thousands, maybe tens of thousands of labs, and everybody's sharing notebooks and equations and tools and models and data and uploading data. You try and build a brain region that doesn't have enough data, somebody starts saying, oh, this is silly, you know, they don't know about this type of cell. So upload the data and then it gets better and better. So I'm very, very excited about it. And I'm really happy and grateful for the long support the EPFL gave and the support they gave us to actually launch also the Open Brain Institute as a foundation so that it can really be dedicated to helping the community work together to advancing brain research.
Wonderful. Thank you for those closing remarks. I'd like to thank everyone, all our panelists today. So Henry Markram, Polina Shichkova, Dan Keller, Jay Coggan, Mark Mattson, and Pierre Magistretti. Thank you all very much for your contributions today. And out there, thank you very much for joining us. I hope you enjoyed the session. If we didn't get to your questions, I'm sorry. Many of them will be in the article, as Polina said. Please visit the Frontiers in Science website to read the article we've talked about today and its related content. You can continue the conversation using #FrontiersForum. Please subscribe to the Frontiers in Science updates. There'll be a link in the chat, and visit the website for upcoming work. For now, from my guests, from everyone, thank you very much. Enjoy the rest of your day. Bye-bye.
Thank you.
Bye.
Bye.